So when we think about the genetic variants that have been linked to different diseases, we often ask, well, if they don't affect proteins, what are they affecting? And the conclusion has been that they have a variety of effects. Some affect things like splicing of RNA, but the majority of them seem to be located in gene regulatory elements, so those that control when genes are turned on and off in different parts of the body. And so the localization of these genetic variants to these regulatory elements is a way of trying to understand how they contribute to disease. We want to figure out if they're affecting gene regulatory elements that act in the brain or the blood, for example. And so the interesting sort of kind of by accident finding that we had just by what I call low throughput bioinformatics, which is manually looking at some of the results, which is literally spending time looking at data sets that describe the genome and scanning through and noticing an unexpected pattern, which was that when there was a genetic variant, it wasn't just overlapping one regulatory element. It was overlapping these massive regulatory elements that, so instead of like one hill, it looked like a mountain range. That's how the data looks. And so you'd see this genetic variant overlapping this mountain range of regulatory elements, and that was not the normal. That was not the normal pattern for these regulatory elements. And when you look at an observation like that, we can statistically quantify it, we can ask it more rigorously, and find that these elements are sometimes referred to as super enhancers or enhancer clusters, but they are where the genetic variants tend to act. And so when I saw that, the genetic variants are localizing to these regions where multiple different regulatory elements are active at the same time, all in the same general region of the genome, that just screamed to me, you have to be looking at how genetic variants act in combination. If one variant is affecting this large region, what about all the genetic variants next door? What are they doing? And so that was sort of the impetus of my graduate work, and it's still what the lab focuses on, is how we can think about how these regulatory elements work together, and in turn how the genetic variants underlying these regulatory elements work together.